Good afternoon. I'm Linda Marban, the CEO of Capricor Therapeutics. I can't tell you how many times I've been at this meeting, and I'm so excited to be in front of real people again. Thank you so much. So my CFO put a lot of slides in here. It's supposed to be a corporate overview. I guess I'm supposed to be really happy that we have so much going on, but I'm going to go really fast, so fasten your seatbelts for a really quick ride. Publicly traded, NASDAQ listed, CAPR is our symbol. This is the manifestation of our filings with the SEC. We have recently moved to San Diego from Los Angeles. We've maintained our manufacturing and quality team in Los Angeles, but most of our work now will be done just down the road from here, and we're really excited because this is because we're expanding so rapidly with all of our programs. We're going to be focusing for a few minutes on each of our programs today. We essentially have two programs with our late-stage cell therapy. CAP-1002 is its commercial name. Its research name is cardiosphere-derived cells, or CDCs. And then we have, in the last few years, moved into the exosomes as a platform technology, both using them as delivery devices for nucleic acids and proteins, taking advantage of our years of experience in cells. And I'll tell you a little bit about that journey in the next few minutes. So let me tell you a little bit about CAP-1002. CAP-1002 is a cell therapy, but it is not a stem cell. So we entered into the arena of cell therapy during the early halcyon heydays where everybody thought they had discovered a stem cell that was going to go in and graft into a tissue and heal it. What we learned in our 10-year or more journey is that, in fact, our cells are not stem cells. What they are some version of a stromal cell, slightly a version of a progenitor cell, very unique. It's found in the heart. We isolate out the heart, the cells from the heart using proprietary methods, and then grow them up to become doses. Our current dosing paradigm is 150 million cells, and most of our therapeutics are repeat dosing 4 times a year intravenously. We have had the cells in over 200 people, and there's hundreds of papers validating the mechanism of action of these cells, which I'll talk about in the next few minutes. What I want you to know is that we've spent years building out the platform and understanding the mechanism of action of the cells and isolating it down to really 3 microRNAs that are driving the potency of the cells. More on that later. So let me tell you a little bit about the disease that we're working on in our late-stage clinical trial. We've recently announced a plan for a phase 3, which we will be talking about as I go through this. For those of you that don't know, muscular dystrophy is an X-linked disorder. It means it's primarily in boys and young men, diagnosed around the age of 3, which is when parents start to notice their children losing milestones, and they typically live now until their mid-20s, early 30s with the advancement of good therapeutics, mostly modulation of the immune system through corticosteroids. There's different types of muscular dystrophy. The most severe is Duchenne muscular dystrophy, the one that we're working on. It means the mutation does not allow translation of that protein, which acts both as a cushion and as a glue to protect the cell from denigration. So we are focusing on the later stage of Duchenne. Let's see if I can make this pointer work. He told me everything else. Yay. So we are focusing on the later stage. Our patients are primarily those ones that have lost ambulation. That means they're dependent on a wheelchair for most of their activity. They have about half their lives ahead of them now with the advent of steroid therapy and artificial ventilation. So we're looking at a 15 to 20-year treatment paradigm, and these people are alive and want to continue living, and we are focusing primarily on the improvement of cardiac muscle as well as skeletal muscle function. So keep in mind these are mostly patients that are off their feet. We had a positive phase 1 clinical trial. It was called Hope Duchenne. In the interest of brevity, I'm not going to go through that data, but it was published in the Journal of Neurology several years ago. We used that study to jump off of and design what we called the Hope 2 clinical study. It was phase 2, obviously, at 9 sites across the United States. We enrolled 20 patients total before we unblinded to see if we had a clinical efficacy, and the data was really very strong, and I'll show it to you in just a moment. Whoops, I'm showing it to you before I wanted to. Well, you're going to see it now. So our mean age of our patients were 14 years of age, and that means they were off their feet. We had to use a measurement of skeletal muscle function that took advantage of the fact that they still had primarily upper body function, right? They couldn't do a North Star ambulatory. They can't do a 6-minute walk test. So early in our clinical development program, an assay came out called the Performance of the Upper Limb. The PUL is sort of what it's commonly called. We're now into the version 2.0, but when we did our clinical trial, we had the 1.2. And what you see here shown is the data shoulder shoulder, arm, and hand, but what you're seeing now is the mid-level, which is the arm. It was the primary efficacy endpoint of the trial. We show a 71% slowing of the disease process using CAP-1002, delivered 4 times quarterly. It's a simple IV infusion. Ultimately will be done in an outpatient center. And you can see in this bar chart here that we had statistical significance. So this is the pooled data that you can see here. Really exciting data. We took it to FDA, and they have encouraged us to do a phase 3 clinical trial based on the pull. And then what is most extraordinary is the cardiac function data. So that's what you see here. This is ejection fraction shown here. It's the most commonly used measure of how the heart meets the needs of the body. Look at the statistical significance of P002. This was measured by MRI and by a core lab, so there's no way that you can sort of subjectively determine whether the heart is functioning better. It absolutely is in the treated patients. And then we have a biomarker called CKMB. It's a fraction of total CK. It's one of the biomarkers that's commonly used if they think you might be having a heart attack. They can take a sample of your blood, look at it, and determine if that's elevated. It means that there's cardiac muscle breakdown. What we see here is that in the CAP-1002 treated patients, and this is after a year of therapy, they have significantly less CKMB, significantly less breakdown of cardiac muscle, supported by the improvement in the ejection fraction, right? So you have a measure of clinical as well as a biomarker of improvement that is not seen in the placebo patients who continue to have elevation of CKMB above baseline, and this is all change from baseline. So we went to FDA and we talked to them with our RMAT designation, our orphan disease designation, and all of the things that you're supposed to get preferred access to, but basically they still wanted us to do a phase 3 clinical trial. They felt that the sample size of 20 patients was too small. So we've designed a phase 3 clinical trial. We're ready to go as soon as we identify the appropriate partner to roll that forward with. We have approval of our protocol and our IND from FDA, so we're enroll the patients the day the partner walks through the door. Oops. I am bad at this thing. It's a phase 3 trial, as I mentioned, all the bells and whistles, 65 to 75 patients, so it'll probably be around 70 patients. We're estimating 20 sites in the U.S. We're going after the same patients. We're going to be using the Performance of the Upper Limb 2.0. This was requested by the FDA that we use the new version. It's like your smartphone. It's the newer, better version of the Performance of the Upper Limb and encompasses the shoulder, the arm, and the hand. I didn't show you the data here today, but that was also statistically significant in HOPE-2. And obviously, we're going to follow up with the secondary endpoints of cardiac function, quality of life, and clinical status. We also, and it's available on our website, we couldn't show them today, but we recently showed a video of one of our patients at the Parent Project for Muscular Dystrophy meeting. It's on my website. Go take a peek at it. It really is extraordinary when you see this young man who was unable to sit up in bed, able to sit up and lean forward after just 12 weeks after his first treatment with CAP-1002. So we're getting a lot of pressure from the community, and because of that, we're continuing our open label extension and expanded access programs. Okay, so let's talk about COVID. Let's go from, one bad disease to another. So I love this slide because it really sort of identifies sort of the phases of COVID. You have the viral phase. It's the phase that, most people get sick, tell their war story, and recover from. Some percentage of people end up with problems breathing. They end up going into the hospital. They need supplemental oxygen. And if we can't pull them back from the cytokine storm and the hyperinflammatory phase, they end up on a ventilation. What we found is that this phase, the phase shown in yellow, is the phase at which you have the most opportunity to help people. We decided to treat patients with COVID during the early days of the pandemic when everybody that had something that was anti-inflammatory were sort of seeing what they could do. We published our work in basic research in cardiology of 7 patients, and what we found in those case studies is that if we treated those patients in that sweet spot, in that yellow phase, they actually got better. If they were already ventilated, multi-weeks on ventilation, unfortunately the disease process had taken over their body and we couldn't pull them back from the edge. So we are finishing up a 60-patient, also randomized double-blind placebo-controlled trial. We call it phase 2, but most of the endpoints are exploratory. Most of them are based on, time in hospital and all that other stuff. We'll expect to have this data out soon. This is one dose of 150 million cells, and it's being done at 4 sites in the United States. So look for that data coming up soon. It really is going to be a validation of the clinical aspects, efficacy of CAP-1002. Okay, are you tired yet? I'm almost tired. Okay, so let me talk to you a little bit about our exosome platform technology. So, exosomes are the words of cells. I bet everybody in this room knows what an exosome is, but I do a lot of investor presentations. So for those of you that don't, basically they are nanometer-sized lipid bilayer vesicles that are packed with mostly nucleic acids, proteins, other types of communication devices between cells, just like our words are to each other, and they go back and forth between cells. We discovered exosomes as the mechanism of action of our cells a few years ago. That's how we got to those microRNAs. And what we ultimately decided is that, well, we're very interested in the exosomes made by our cells. It really is almost like, Regen Med 2.0, right? Passive activity of a cell that you already know can work. Why not just use the cell? So we decided to really walk hard and fast into engineering the exosomes to deliver primarily nucleic acids and proteins. And so what physiologic problems can we address using exosomes? Well, I think everybody knows that gene therapy is, really exciting in terms of its opportunities, but also has a lot of safety risks that are still associated with. We can potentially address some of those just using exosomes as nature's delivery vehicle. Let's get past that cell membrane and cause a change in transcription and translation. You can deliver RNAs. That means siRNAs potentially for oncology. That means that you can use microRNAs for transcriptional regulation. And obviously, and we all know now, and it's funny because when I first started talking about exosomes, pre-pandemic, nobody knew what a lipid nanoparticle is. I bet my high school student knows what a lipid nanoparticle is now. But we also know that they have safety risks, and there's a lot of data coming out about myocarditis that may be caused by, the inflammatory response of these lipid nanoparticles. So exosomes really can target all of these issues, and we have a fully engaged team of 14 PhDs and scientists right down the road in Torrey Pines working on the engineering of the exosomes now. The first thing we wanted to do is see if we could get expression of a protein, and what we show here, and I can't go into the details because I'm running out of time, is if we use this marker called antares, which lights up. It's an enzyme, so basically you put the mRNA for antares in the exosome, you inject it in the little mouse, then you take them and you put the little enzyme in there and the buffer, which you inject them with, and then as the protein has been translated and the enzyme is cleaved, you get it to light up. And you can see we get nice bright signals. You can also see that you have some really dense signaling in areas where you think you'd see it, right? The liver, potentially, the spleen, that area there. So now we know we can deliver an exosome loaded with an mRNA and get protein expression and, even more importantly, enzymatic activity. It's a functional protein. So we started working on an exosome-based SARS-CoV-2 vaccine. We started a while ago. It takes a little bit longer because we're working with a brand new delivery device. We've met with FDA. We anticipate putting out an IND for this clinical trial. It's a bivalent vaccine. We started multivalent, have narrowed it down to the two proteins that are most active. This is the N, the nucleocapsid, and this is the S. Everybody knows the S. The N is the most highly conserved of the COVID proteins, so we really think that this will be mutation resistant. We're getting really good data, which I can show you here. This is the antibody data, but for the anti-N and anti-S, it's after several vaccinations and out to 84 days. This is the T-cell response. This is, the money shot, really, because this is, the memory response. And again, we're getting memory to the N and to the S. This data was recently published in the Journal of Biologic Chemistry. I can provide you with the manuscript, or you can find it on PubMed. But memory response, antibody response, and we're taking this bivalent vaccine made with exosomes into the clinic. It also opens the opportunity to do bivalency with other types of infectious diseases. So why not a flu plus COVID vaccine sometime in the future? All right. In addition, we're working on several other opportunities. I mentioned, pick your nucleic acid of choice, and we can go into it and explore it. And so we have a big pipeline in the lab that we haven't disclosed yet, but you'll be seeing news coming out soon. If you're interested in partnering and developing an exosome-based therapeutic, we're very interested in talking to you. This is our milestone slide. I did it. And this is my team. So I'm Linda Marban. I'm the CEO. I'm supported, and very lucky to have Christy Elliott have recently joined us. She came to us with many years of exosome experience at Hopkins Ph.D. Dr. Steve Gould is our executive consultant. He's the founder of the American Society of Extracellular and Microvesicles, so he knows a lot about exosomes. And then, of course, my team at home, who said I could do this in 15 minutes, and I did. So I think I even have time for one quick question. Does anybody have any questions? Okay. I look forward to seeing you out and about and in the halls, and hope to get in touch with you soon. Thank you so much for your time.